Hello guys, welcome back to my channel. So let's get started with today's topic, that is transducer mechanism and G protein couple receptor. Starting with, what is transducer mechanism or signal transduction? Simply we know that receptors are present on the surface of our cell. So whenever we are taking a drug and this drug is combining with the receptor, it is going to show a response. But the question is, how is it going to show this response? The simple answer to it is through the transmission of the signal. So signal transduction is majorly grouped into 5 categories, that is G protein couple receptor, ion channel receptor, transmembrane enzyme linked receptor, transmembrane JAK-STAT binding receptor, receptors regulating gene expression or the nuclear receptor. So here we have a structure of a G protein couple receptor where we can see 6 loops. 3 are lying on the extracellular region and 3 are lying on the intracellular region. Gpcr receptor is also known as heptahelical or transmembrane receptor. As we can see, here we have 7 helices. Also, we have at this position heterotrimeric structure where alpha, beta, gamma are the components. Whenever this GPCR receptor is in inactive state, this is the configuration where with this heterotrimeric structure with the alpha, GTP is attached. This is the inactive state. But whenever any agonist combines with the receptor and signal transduction starts, it gets activated and the change is GTP replaces GDP over this position and alpha gets detached from its component beta and gamma. Whenever alpha is getting detached, it goes and shows its effect on the effector and shows a response. After it has completed showing its response, it again comes back to its original position with gamma and beta, and the GTP that was present with alpha gets hydrolyzed to GDP again. For the alpha to show its response in the effector, usually it follows 2 paths, that is, it activates the adenyl cyclase and follows the CAM pathway, or it can activate the phospholipase C and follow the IP3-DAG pathway. And for beta and gamma, they directly controls the channel regulation. So we will see what happens when the alpha at this position gets combined with the receptor, and if the receptor is coupled with these G proteins. These are the subtypes of the G protein. So whenever the receptor is coupled with GS, which is stimulatory in nature, it will activate the adenyl cyclase and also it can directly open up the calcium channel. Or if the receptor is coupled with GI, it is inhibitory in nature, so it will inhibit the adenyl cyclase or it will open the potassium channel. If the receptor is coupled with G0, then it will inhibit or close the calcium channel. Or if it is coupled with GQ, it will activate phospholipase C. Now, for example, the alpha has activated the adenyl cyclase, then it will follow the CAM pathway. So after it has activated adenyl cyclase will convert ATP to CAMP. This CAMP is a secondary messenger. This CAMP will in turn activate phosphokinase. This phosphokinase will phosphorylate the enzymes. So what is phosphorylation? Basically, phosphorylation is a process where a phosphate group gets added to any organic compound. For example, if we have ADP, adenosine diphosphate, it will get converted to ATP. Previously in ADP, we had 2 phosphates. After phosphorylation, it will have 3 phosphates. ATP, adenosine triphosphate. So this was a simple example of phosphorylation. So after phosphorylation of enzyme occurs, these are the following responses that can be seen. First, increased contractility, then relaxation of smooth muscle, glycogenolysis, lipolysis, inhibition of secretion, water conservation by kidney. Also, the CAMP can directly open up the calcium channel cyclic nucleotide gated channel, which will in turn increase the cardiac contractility. So, all this happens only if the adenyl cyclase gets activated. And for adenyl cyclase to get activated, the receptor should be bound with GS. And if the receptor is bound with GI, which is inhibitory in nature, it will inactivate the adenyl cyclase and all these sequences will not occur. And this CAMP, which is a secondary messenger, it will get inactivated by the action of enzyme phosphodiesterases that will decompose or degrade CAMP to 5-AMP. So this was about CAMP pathway. Second pathway that it can follow is by activation of phospholipase C. So after phospholipase C is activated, it will hydrolyze the membrane phospholipid phosphatidylinositol 4,5-bisphosphate, or also known as PIP2. Then it will generate inositol 1,4,5-triphosphate, which is also called IP3, and diacylglycerol, also known as DAG. So this IP3 and DAG, they are also secondary messengers. Here, IP3 is water soluble in nature, whereas DAG is not water soluble. Instead, it is lipophilic in nature. So because IP3 is water soluble, it will easily diffuse into the cytosol, whereas the DAG, it will stay in the membrane only. Now, we will see what happens when IP3 gets diffused in the cytosol. After getting diffusing, it will start mobilizing or collecting calcium from endoplasmic reticular depots. This released calcium are very versatile regulators and they will start mediating some action, like it will cause smooth muscle contraction, glandular secretion or release, cell proliferation, etc. And this signaling of IP3 will get terminated by degradation of the secondary messenger, which is this. After it gets degraded, all these actions will stop. Now we will see in case of DAG. DAG is lipophilic in nature and they will stay in the membrane only. While staying there, they will start recruiting protein kinase C and they will activate it with the help of calcium. Now the activated phosphokinase C start phosphorylating many intracellular protein and they will mediate the physiological responses. So even they will start showing some of the responses. All these actions will get stopped whenever these IP3 and DAG, which are the secondary messenger, will get terminated. And after IP3 it will get terminated, it will produce inositol again. And this inositol will again be reuptaken for the formation of PIP2. And after DAG is get terminated, its metabolite are phospholipids and arachidonic acid. This was about the IP3-DAG pathway. The last is the channel regulation. In case of channel regulation, earlier we have seen when the alpha is going to show its effect on the effector, the alpha is going to regulate the activate the adenyl cyclase pathway and the phospholipid C pathway. But the beta and gamma, they can directly do the channel regulation. They will control this channel regulation. In this case, they can open any channel without the intervention of the secondary messengers. Now, secondary messengers we have seen in case of phospholipase C, the IP3 and the DAG, and in case of adenyl cyclase, the CAMP. These are the secondary messengers. But in case of channel regulation, these secondary messengers are not used. They can directly open up the channel by the help of these G proteins. So the receptor, whenever it is coupled with GS, it will open up the calcium channel directly in the myocardium and the skeletal muscle. If it is combining with GI and GO, then it will open the potassium channel. And if it is coupled with G0, then it will directly inhibit the calcium channel. So this direct channel regulation is done by beta and gamma subunits. So that's it for today. I hope this video was helpful for you. If it was, then please don't forget to subscribe to my channel and let's meet in the next video. Thank you.